Hello, everyone. My name is Frank Jenkins, and my capstone project for the Springboard Data Analysis course with Python looked at differential expression and co-expression analysis of kidney cancer cells versus expression values for normal cells. The data set was downloaded from the Cancer Genome Atlas, which is managed by the National Institute of Health and the National Cancer Institute. Just a brief background on the Cancer Genome Atlas. It is a resource that is very useful to the cancer research community. It contains gene expression data sets and other types of data sets for 33 different types of cancer. 10 of those are rare cancers. Um, and just want to briefly talk about the cancer that we studied in this case. Okay? It's called clear cell renal carcinoma. It's a form of kidney cancer which primarily afflicts older men. It's called clear cell because the cells generally have a clear cytoplasm surrounded by a distinct cell membrane containing round and uniform nuclei. And just as an FYI, our cytoplasm, okay, is the space in our cell, inside of our cells, that lie between our cell membrane and the membrane that surrounds our nucleus. Um, Now, in general, renal cell carcinomas are a kidney cancer that originates in the lining of the proximal convoluted tubule, which is a part of the network of small tubes in our kidneys responsible for transporting urine. Initial treatment for this cancer is generally pretty successful. It involves either partial or complete removal of the affected kidney, and in most cases, at least in stage 1 and stage 2 cases, the 5-year survival rate is very good. It's between 65 and 90%. The greatest risk factors for renal cell carcinomas are generally lifestyle related. There's only a very small incidence of inheritable factors that are linked to renal cell carcinomas. For the most part, it's lifestyle factors that are the most important predictor of this type of cancer. They include pretty typical factors: obesity, smoking, and because we're dealing with the kidney, hypertension is also an important risk factor. Um, so, problem identification and target audience. So the target audience for this would be novice bioinformaticians who need accessible resources to learn programmatic techniques for genomic analysis. Um, it could also be lay people who are interested in understanding risk factors associated with cancer. Um, and what we want to do, the problem that we're trying to address is we want to understand the differences in gene expression levels between cancer cells and normal cells. And that will help us identify the genes that are involved in formation of cancer, in sustaining the tumor microenvironment, and metastasis to other parts of the body. And this, of course, is absolutely necessary when we're trying to develop treatments for cancer and so forth. And also trying to identify risk factors for cancer to prevent it in the first place. Um, and the workflow is a pretty standard data analysis workflow. Of course, we have to find and import our data set. We have to wrangle the data, filter, conduct exploratory data analysis. In this case, we also conducted a correlation analysis, differential expression analysis, and unsupervised learning, which we use hierarchical clustering in this case. And then finally, we close it with a hypothesis test and an interpretation of our results. So the data, as I mentioned earlier, the data was downloaded from the Cancer Genome Atlas as a CSV file. Of course, we had to change the working directory to point at the path where the data set is stored, and then assign the data set to some kind of file name object. in my case, I simply assigned it to an object called file name. And then the data set was imported into a Jupyter notebook using the pandas read CSV command. Finally, the columns containing the G names was set as the index. And here at the bottom of the slide, you can see the URL where the data set can be found. Unfortunately, the data set was too large to upload to GitHub. So, however, I did include the URL both here in this presentation, in my report, and also in my Jupyter notebook containing the project itself. Here is a head of the original data set. Okay. This is just a sort of a sample template for the data preparation workflow. So to wrangle and filter the data, the first thing I did was I dropped all rows with 0 values. And then I split the data frame into two. Okay, the one data frame contained normal cell gene expression values, and the second data frame contains cancer gene cell expression values. The original data set was already normalized and logged to transform, which cuts down on the number of steps needed to pre-process the data. Okay, then I conduct exploratory data analysis. Okay, so the first thing I do is I simply confirm the shape of the distribution, looking at both the data frame with the cancer expression values and the data frame with the normal cell expression values. Okay, and as we see, in both cases, the data appears to be normally distributed. Next, I conduct an analysis to try and sort of just do a preliminary analysis to understand if there are important biological differences between our two data sets. Okay, and this is important because if we don't see sort of easily recognizable differences between our two data sets, it may not be a great data set to explore. And so this scatter plot is a plot of mean gene expression values versus the variance. The second scatter plot is simply a plot of mean transcript levels for normal cells versus mean transcript levels for cancer cells. And we see in both cases there are a lot of points of data that sort of deviate from the majority of the data. And that indicates that there are likely real biological differences between cancer and normal gene expression levels in our data set. Then we conduct differential expression analysis. I use the t-test method in this case. But I should explain there are a number of different methods which can be used for differential expression analysis. They include the t-test method and other methods such as empirical Bayes. Now, the t-test is a good method if you have a large number of samples and the data has been normalized. Okay, and both of those factors are true in this case. However, if you have a very small number of samples, you may want to consider using the empirical Bayes method. And also, empirical Bayes has a number of variations, some of which are non-parametric. So, which of course do not make assumptions like normality. This slide just shows the formula for the t-test. Here we see the top 25 differentially expressed genes. Okay, and I know this may look like gibberish to a layperson, but on the left-hand column contains the actual gene names for our top 25 differentially expressed genes. Then we conduct a correlation analysis. I simply use the pandas correlation method. I generated heat maps showing correlation with our differentially expressed genes. And what we notice, and we'll see these heat maps in just a moment, and what we will notice is that there are a large number of negatively correlated genes in the cancer expression data frame, while the opposite is true in the normal cell expression data frame. And as I explained here, this is not surprising. Oncogenes routinely produce deleterious proteins, which can often inhibit cellular functions like cell cycle control and apoptosis. And just as an FYI, apoptosis simply means cell death. And there are a number of genes within our genome that can actually promote apoptosis. And it's an important function in many cases, especially where you have deleterious mutations that our DNA repair machinery were unable to repair. And then finally, I generate pairs plots just to simply demonstrate or to kind of highlight the trends which appear in our heat maps. Now, this is a heat map of our top differentially expressed genes correlated with our cancer data set. Okay, and you see here a lot. This heat map is mostly dark, which if you look at the legend here, that indicates values below zero. Okay, correlation coefficients below zero. And that tells us that there's a lot of negative correlation in this data set. Conversely, when we look at our normal gene expression data set and correlate that with our top 25 differentially expressed genes, we don't see so much negative correlation. In fact, you see predominantly positively correlated genes. And this is further highlighted by our pairs plots. I just want to say that I was really reluctant to do a pairs plot, but I figured I'd try it anyway. Normally a pairs plot with so many different sort of subplots are very hard to read or even impossible to read. But in this case, I think the plot came out really nicely. You can see the shape of the distribution if you go down the diagonal, and you can see in this case that most of these, many of these genes are negatively correlated, right? Because you see a negative slope in the shape of the data. And conversely, the opposite is true with this. You see mostly positive slopes where you can see a slope. And we can also see the shape of the data here, and we can see the skew in the shape. Okay. So next, we wanted to isolate our top correlated genes. Okay? It's not very interesting to look at genes that only have a sort of a small correlation coefficient. We're really interested in genes that have a correlation coefficient greater than 0.9 and less than 1. Okay, and less than 1 is important to denote because, of course, every gene will be correlated with itself, right? Or the correlation coefficient when you look at a gene's correlation with itself is going to be 1. And this is simply the code I use to do that. And then we move to unsupervised learning. So I just want to briefly just highlight one of the important reasons why I did a correlation analysis in addition to the differential expression analysis. It's always challenging to sort of truncate your data set enough so you can produce meaningful dendrograms in the context of hierarchical clusters. If you have too many genes, you'll wind up with clusters at the bottom where the gene names overlap each other, and the dendrogram is simply not readable and not comprehensible. So it's really important to really try and narrow down the number of genes that you're analyzing and really focusing in on the question or the problem that you're trying to solve, and just eliminate as much of the noise as possible. And so that is one important reason that I did the correlation analysis. Okay, and by correlating with our top 25 differentially expressed genes, I was really able to sort of truncate the data set enough where I could produce nice and readable dendrograms where we can actually identify the gene names and identify the clusters. So you see those clusters here, or I'm sorry, the dendrograms here. And what I focused on and we'll take a look at this other angiogram as well. I focused exclusively on the green cluster. Okay, my goal at the outset was simply to identify one gene, one interesting gene that I could conduct some brief research on, and formulate a hypothesis, test the hypothesis, and briefly discuss and sort of elucidate on for everyone's edification. And here is the way I formulated the hypothesis. The null hypothesis is that ANGPTL4 is not overexpressed in clear cell renal carcinoma tumor cells relative to expression levels in normal cells. The alternate hypothesis is that ANGPTL4 is indeed overexpressed in kidney cancer cells relative to normal cells. Okay, and as we see, our p-value is extremely small. It's I'll just round it to 2.88, raised to the negative 89. As you can imagine how tiny that number is. It's obviously far below 0.05 or the significance level. So this provides strong evidence against the null hypothesis, allowing us to reject the null hypothesis and assert that ANGPTL4 is indeed overexpressed in kidney cancer tumor cells compared to normal cells. Okay, and here I include an image that shows the role ANGPTL4 plays in tumorigenesis or cancer formation. Okay, now this is an image showing its role in epithelial cells, not kidney tissue, but nonetheless it gives us some information. So as we can see here, the gene in one instance, it's used in wound repair, okay? But in another instance, it can also promote tumorigenesis, okay? And in fact, it can also promote angiogenesis and metastasis. Now, just to briefly explain what angiogenesis is. Angiogenesis is the process by which the tumor microenvironment forms its own blood vessels, okay? Like our normal cells, tumor cells also need access to blood so it can derive oxygen. Now, I should also mention that angiogenesis in the tumor microenvironment is normally a pretty crude process, and you get a network of these small capillaries that aren't very efficient at delivering oxygen to cancer cells. So you see many cells that generate energy via the fermentation pathway versus the normal pathway. And this is why the tumor microenvironment often has a slightly lower pH compared to our normal physiological pH. In other words, it's slightly acidic. And metastasis, of course, is simply cancer spreading to other parts of the body. This image shows something that I find pretty interesting. It shows that our gene of interest can play a dual role. It can both promote tumorigenesis and it can also prevent cancer, or it's at least helpful in inhibiting tumorigenesis. And that's pretty interesting, although I should mention that there are many, many genes that have a similar sort of dual role in the context of cancer. In conclusion, as I discussed earlier, we have a very small p-value, significantly below our significance threshold, so we can reject the null hypothesis and assert that ANGPTL4 is overexpressed in clear cell renal carcinoma cells. There's one important thing I want to note. Although there has been extensive research elucidating on the role ANGPTL4 plays in tumorigenesis and functions like glucose homeostasis and insulin sensitivity, we were able to identify ANGPTL4 with no prior knowledge. In other words, I did not go into this analysis searching for correlations or clusters involving ANGPTL4. Okay? It simply came up during the course of the analysis. And ultimately, I sort of, it was somewhat of an arbitrary choice to pick ANGPTL4 for further analysis, but it made sense for a variety of reasons. I also want to just kind of briefly just again highlight why this project is important. Okay, first off, it demonstrates techniques to conduct a basic gene expression analysis. Okay, I also discuss many alternative techniques, and I show how to actually code for those alternative techniques in my final report. Okay, so if anyone's interested, the final report is on my GitHub page. But this also shows how open-ended data analysis can both spearhead biological research and hypothesis development. And this is really important because, with sort of shrinking funding for scientific research, um, you know, getting a grant has just become ever more competitive as time goes on, and it's very important to be able to provide statistical evidence that you are likely to make a biologically important discovery right at the culmination of your research. So bioinformatics is becoming increasingly important in this context, um, and and I guess I should also discuss some recommendations and sort of where to go with this research or next steps. So in a more typical analysis, we wouldn't just focus on one gene, right? We would want to analyze all of the genes that appear in our cluster maps or in our dendrograms, okay? We would, and ideally, we would want to find a gene that has not been extensively studied, or at very minimum, we would love to find a gene that has not been studied in the context of what we're actually looking at. In other words, in this case, it's kidney cancer. Okay. When we find a gene like that, and if we're fortunate to find a gene like that vis-a-vis data analysis, the next step would be to conduct wet lab studies. Okay, so we would want to really try to understand what pathway that gene functions in, what is its what is its role, what kind of protein does it produce. What are what transcription factors promote its transcription or inhibit its transcription, and et cetera, et cetera. So that would be, and I include, by the way, 3 specific recommendations, which also, by the way, includes a sort of a population statistics analysis to better understand the role between obesity and kidney cancer. Okay, so as I discussed very briefly earlier, but also extensively in my report, ANGPTL4 plays a number of roles in our bodies. It is responsible for, or it plays a role in glucose homeostasis, insulin sensitivity, and again, it also has a role in tumorigenesis as well. But it sort of begs the question, given the role of AMG PTL4, okay, and also the connection between AMG PTL4 and obesity, what, and also, excuse me, I should say, given that we also know that there's a strong connection between obesity and the risk of kidney cancer, okay, along with other factors like hypertension. But obesity is one of the big sort of predictors for this type of cancer, and quite frankly, most types of cancer. But given that, and given the role of ANGPTL4, might it make sense to try and really understand, is it just obesity, or does sugar consumption play a role? Again, because ANGPTL4 plays a role in glucose homeostasis and insulin sensitivity. Okay, so might high carbohydrate and sugar consumption increase our risk of cancer, or of kidney cancer in this case? So Anyway, with that, I will close, and I want to thank you for listening to this presentation, and I hope you have a wonderful day.